Hi, welcome to Physionic, where we learn the body from the macro to the micro. If that's something you think you'd be interested in, then consider subscribing. So today's video is going to be discussing the physiology of muscle hypertrophy. How do our muscles grow, essentially? And I released a video on this a few years ago, but I'm updating the video based on new research that has come out. So now the body of research is pointing towards a little bit different conclusion than what I had in that previous video. So I'm updating, and I've also updated the article so you can check that out for far more details. So without further ado, let's go ahead and jump into it in a short, sweet, science-based video. Let's get into it. 2 years ago, I released a video on the physiology of muscle hypertrophy based on 17 references from various universities and studies. Now we are revisiting the subject because a new series of studies gives compelling evidence that the previous train of thought, muscle microtears is the primary driver of muscular hypertrophy, is incorrect. Muscle hypertrophy, for a long time, was considered to be a product of muscle cell microtears, as the sarcolemma of the cell, as well as the contractile units within the cell, were damaged from the exertions of training.
When an athlete engages in a heavy workout, this structure is damaged, causing microtears in the muscle fibers. The body naturally repairs this damage by mending the torn fibers. The result is a muscle that's larger and stronger than before.
However, while this physiology is correct, these microtears may not be the primary driver of muscle hypertrophy. Instead, mechanical tension and metabolite formation are likely superior players, especially the former. Mechanical tension is the passive stretch of the muscle and the contraction creating active tension to resist and move the weight in a different direction than it would, uninhibited. This stretching and flexing is reflected on a cellular level as the membrane of the cells must stretch and shorten accordingly. This action alone is thought to induce muscle hypertrophy, independent of muscle cell damage. How it does this is via a series of different pathways. Some will be detailed now. One, as the neuron fires to tell the muscle to depolarize and contract, there is a release of calcium from an organelle known as the sarcoplasmic reticulum. This calcium is a key to muscle contraction, but also binds to a protein called calmodulin. Calmodulin then binds to a protein series which eventually activates a well-known protein, AKT, which then activates the master protein synthetic stimulating mTOR, and further downstream protein synthetic transcription. The second mechanism is through the translocation of proteins related to titin in the sarcomere of the myofibril that move from a complex near titin to the nucleus to initiate protein synthesis. Third, the upregulation of the myokine insulin growth factor, but appropriately classified as a mechanogrowth factor, leads to an autocrine, meaning it acts on the same cell, and paracrine, meaning it acts on nearby cells, activation of AKT, and the stimulation of protein synthesis. Finally, the last point discussed in this video is a brilliantly designed housing of an enzyme by the name of phospholipase D, hence called PLD, which is purposefully separated from its substrate it uses to synthesize another molecule. However, when the membrane is stretched and disturbed, PLD is free to interact with the substrate and produces phosphatidic acid, which binds to downstream protein synthetic transcription factors independent of mTOR initially, then binds to mTOR and potentiates the protein synthetic response further. So in these ways and more, protein synthesis is stimulated for a hypertrophic-like response. Now, Metabolite accumulation also seems to have an effect, and here are a few ways that it too increases protein synthetic response. 1. The accumulation of lactate, hydrogen ions, inorganic phosphates, among other molecules, leads to a greater expression of insulin-like growth factor, which increases the proliferation of satellite cells. I'll discuss those in just a minute. And also increases protein synthesis via similar pathways outlined before. And secondly, through other myokines and hormones like growth hormone lead to increases in interleukin-6, which also has a recruiting effect on satellite cells. Finally, reactive oxygen species, cell swelling, and hypoxia can also affect gene transcription in favor of protein synthesis. Clearly, both of these systems play a role, and likely major roles. But then, where does that leave the microtear hypothesis? In a rather useful place actually. With resistance training, there's going to be cellular damage, and one of the ways to restructure and repair the muscle cells is through the recruitment of immune cells that further release cytokines like interleukins, insulin-like growth factors, and tumor necrosis factors, as well as phagocytose broken parts of the muscle cell. Muscle damage may also play a role in the satellite cell recruitment, as the damaged cells allow areas for the satellite cells to sink into and fuse with, donating their nuclei to increase the protein synthetic capacity of the cell chronically. All in all, muscle damage, if it is merely a byproduct of the resistance training and the two dominant hypertrophic mechanisms, or if it does add its own hypertrophic effect, has a place in the process, just not at the front of the line, as we once thought. So there we have it. If you'd like to actually see more details, you can also check out my old video that I have, which is called The Physiology of Muscle Hypertrophy. I'm going to keep that up, mainly because all the physiology explained within it is correct, and it's got a bunch of diagrams. It's a much longer video. It's like 30-40 minutes, something like that. I go into far more detail with it. That's when I would really go into detail with a lot of my videos. But if you just want to read through it and read through the references that I'm talking about when I talk about all these different things, you can check out the article that I wrote down below that I revamped and re-updated with I think like 9 or 10 more studies, so it should be a grand total of 26 or 27 different studies that I looked at. And with that said, I hope that I have the pleasure of seeing you in the very next video. Have a good one, guys. See ya.